I'm David Fields. I'm a zooplankton ecologist. I've been here for about 14 years, and I study all aspects of how a zooplankton lives in the environment. So I look at the individual levels. I'm interested in the mechanisms that control how they behave. I'm interested in the larger scale processes, like how they interact with the global environment and the effects on them, and also in the trophic transfer. They're a primary food source for fish and transferring that all the way up into the food web. I'm not sure if my videos are going to work, but I'm going to try here. You know, when we got called on to put together our favorite project, that was like Sophie's choice for me. We have so many things going on in the lab that I'm really interested in. I just want to highlight a few of them, and then I'm going to talk about the one that I had to pick. I'm doing a lot of ocean acidification experiments. We're looking at the effects on a calinus species out here and an estuarine species out here and calcifiers, so we're doing that. I'm just writing a proposal today about looking at juvenile lobsters and potentially the effect of ocean acidification on juvenile lobsters. I still do sensory work. I'm interested in how the little hairs on the antenna, which work just the same way as our ears work. I'm interested in looking at how the animal processes information, how the brain processes. So we just had a paper come out this year on that. I look at parasites within the gut of the animal. There are parasites that grow inside of these copepods. They stop the fecundity, which means the population growths slow down, which has implications for how larval fish are going to be able to make it on those kind of food sources. And then I've been doing experiments 2 weeks ago on their ingestion of plastics. And so I'm looking at plastic beads, and I'm tracking plastic beads through the animals. Plastic beads tend to accumulate toxins on them. They also slow down the digestive process, and so we've been looking at the accumulation of those beads inside of the copepods, and that'll be transferred up the food level. I'm doing pretty good here. So what I'm going to highlight here is some work that I've been looking at the biome. So David talked about the biome. Copepods also have a biome. There are about 100 trillion billion copepods out there. If you string them together, mouth to anus, it's like a light year of copepods. So with that, they're all sitting out into the middle of the ocean. Here's my little copepod here, and you can see them, although you can't, sucking down all of the ocean's water through their body. What their gut looks like matters, because essentially you're taking water from the environment, you're passing it through the gut, and how it processes that matters. I'm going to show you just a quick video. I don't know, can you see it here? So we know very little about the copepod. I won't have time. We know very little about the copepod. And just like if you went into the ocean and you found a hydrothermal vent, what you would do is you'd gather some of your favorite friends, they'd develop a probe, you go down there and you examine that hydrothermal vent. I do that with the copepods. I've been developing a probe. I stick it up the anus. I'm the only copepod proctologist, I think, out there. And I examine the gut of the copepod. I'm interested in what bacterial colonies live inside of there. I'm interested in the pH. I'm interested in the oxygen sensors. And we've been doing that. We've been measuring the pH down here and the oxygen within the gut. Am I done? And that is what I do all day.